Welcome to the deep dive. We sift through the sources, find the core insights, and bring them straight to you. Today, we're tackling a topic that, well, impacts almost everyone. Cancer.
Mm.
The numbers are quite stark, around 10 million deaths every year. And for a long time, our main tool, chemotherapy, has been, well, a bit of a blunt instrument.
It really has. Powerful, yes, but not very selective. It often struggles with delivery, getting enough drug to the actual tumor, and then there's the whole issue of multi-drug resistance.
Exactly. It's a huge challenge. We really need smarter, more targeted approaches.
We absolutely do. The biology is just so complex, different tumors, different patients.
And that's where our deep dive leads us today. Our sources are pointing towards something potentially transformative, calling it a new era of precise and personalized cancer therapy. We're talking about smart nanoparticles.
Yeah, and you can really think of them like a super versatile toolbox. That's the analogy we'll use.
A toolbox. I like that.
So our mission today is basically to unpack that toolbox. We'll look at what these smart nanoparticles could do. The potential is huge. But also, importantly, the challenges, the roadblocks stopping them from being everywhere right now.
And the research pushing things forward, too, right?
Exactly, where the science is heading.
Okay, let's unpack it. Smart nanoparticles.
Yeah.
I mean, nanoparticles are tiny, obviously. But what makes this specific type smart?
That's the key question, isn't it? It's not just about size. A smart nanoparticle is more like an intelligent system. It's designed to actually respond to things in the body,
Respond how?
to specific biological cues, or maybe we guide them externally so they're not just floating around passively. They're engineered for precise delivery. Think less random mail, more guided drone.
Okay, guided drone. So what makes an ideal drone? What are the basics for this nanoparticle toolbox?
Right. Well, for this toolbox to work well, you need a few things. A stable size, small nanometer scale. You need to be able to adjust the surface charge that affects how it interacts with cells. High capacity to actually carry the drug, the payload. And critically, it has to be biocompatible, low toxicity to healthy parts of the body. That's paramount.
Maximize the hit on the tumor, minimize collateral damage.
Right.
Makes sense. So how do they get this intelligence? Is it built in or added on?
It can be both, actually. Some materials just naturally have responsive properties. Think certain gold nanoparticles, they react to light or heat.
Interesting.
But more often we engineer it, we modify the surface, attach specific functional groups that react to certain triggers.
Triggers. Okay, so they need something to set them off to release the drug. What kind of triggers are we talking about?
Good question. There are basically two main types. Internal ones, things already present in the body, endogenous stimuli, we call them.
Right.
And then external ones, exogenous stimuli, things we apply from the outside, which gives us more direct control.
Let's start with the internal ones. What signals inside the body can these nanoparticles listen for?
One of the big ones is pH. You see, the area around a tumor is often slightly more acidic.
More acidic.
Yeah, maybe pH 6.5 to 7, whereas normal tissue is more like 7.4. And inside the tumor cells, in compartments like endosomes or lysosomes, it gets even more acidic, down to pH 5 or even 4.5.
Wow, okay.
So this natural pH difference, this gradient, can act as a really neat trigger. The nanoparticle is stable at normal pH, but releases its cargo in the acidic tumor environment.
That's clever, using the tumor's own environment against it. What else internally?
Enzymes. Tumors often have unusual levels of certain enzymes compared to healthy tissue. Things like MMPs, matrix metalloproteinases, they're involved in cancer spreading, or cathepsin B, urokinase, lots of examples. So you can design nanoparticles that are broken down or activated only when they encounter these specific tumor-associated enzymes.
And I've read that tumor cells are under more stress, like oxidative stress. Can that be used?
Absolutely. That's the redox environment. Tumor cells often have much higher levels of antioxidants like glutathione, GSH, sometimes 4 times higher than normal cells.
4 times.
Yeah. So you can design nanoparticles with chemical bonds, say disulfide bonds, that break apart specifically in this high GSH environment, releasing the drug right inside or near the tumor cell, another internal trigger.
So those are signals the body provides. What about the external ones, the ones we control?
Right, these give you that on-demand release. Temperature is one. Tumors can sometimes be slightly warmer, maybe 42 degrees Celsius compared to the normal 37.
Just a few degrees.
Yeah, but it can be enough to trigger specially designed thermoresponsive systems to release their payload.
That seems subtle, but potentially very effective. Light seems like it would offer really precise control, though.
It really does. Light, especially near-infrared or NIR light, because it penetrates tissue better, is a fantastic external trigger. You can incorporate photoresponsive elements into the nanoparticle.
So you shine a light.
Exactly. You shine a specific wavelength of light on the tumor area, and it causes a chemical change. Maybe it breaks a bond. Maybe it changes the particle shape, releasing the drug exactly when and where you want it. Very precise spatial and temporal control.
Like a remote control for drug release. Any other external forces?
Yep. Ultrasound and magnetic fields are also being explored quite a bit. Ultrasound can trigger release, and it can also temporarily make tissues more permeable, helping the nanoparticles get deeper into the tumor.
Ah, okay.
Magnetic fields can be used to guide magnetic nanoparticles to the tumor, hold them there, or even generate heat to trigger release or kill cells directly. And often, these external methods can also help with imaging, like MRI. That combination of therapy and diagnostics is called theranostics.
Theranostics. Got it.
Yeah.
Okay, so nanoparticles can be smart. They can respond to triggers. But how do we make sure they actually get to the tumor?
Yeah.
That guided drone needs a GPS, right? How does the smart missile find its target?
That is absolutely critical, and it brings up a really important point from our sources regarding something called the EPR effect, enhanced permeability and retention. For years, the idea was that nanoparticles would just naturally build up in tumors because tumor blood vessels are leaky and don't drain well. Passive accumulation.
Made sense, sort of.
It did in theory, and it worked okay in mouse models. But, and this is a key takeaway from the research, that EPR effect in actual human patients is highly variable, much less reliable than we thought.
So just relying on leaky plumbing isn't good enough for humans.
Pretty much, yeah. It's too unpredictable. We can't rely solely on that passive approach for the kind of precision we need.
So we need that active, missile-like guidance system. How do you build that?
That's where active targeting comes in. We basically decorate the surface of the nanoparticle with specific molecules, tumor-specific ligands.
Ligands.
Like little keys. Exactly like keys. Keys designed to fit specific LOX receptors that are found much more often on cancer cells than on normal, healthy cells.
What kinds of keys are we talking about? Give me some examples.
Sure. A classic example is antibodies. We can attach antibodies that recognize proteins commonly overexpressed on cancer cells, like EGFR or HER2, which you see a lot in breast cancer,
Right.
or PSMA for prostate cancer. These antibodies basically act like homing devices, guiding the nanoparticles straight to the cancer cells.
Antibodies are pretty big molecules, though, aren't they? Does that cause issues getting into the tumor?
That's a very sharp point.
Yes, their size can sometimes limit penetration, so researchers also use smaller keys, like peptides.
Peptides.
Yep, short chains of amino acids. They can be designed to bind very specifically, too. Like RGD peptides, they target integrin receptors involved in making new blood vessels for the tumor.
Cutting off the supply lines, kind of.
Sort of, or using those receptors as docking points. And then there are really cool ones called cell-penetrating peptides, CPPs, like TAT peptide. These can actually help sneak the whole nanoparticle inside the cancer cell.
Whoa, okay. Breaking and entering, basically.
In a therapeutic way.
Yeah.
And then there's another class, not protein-based, but using nucleic acids.
Yeah.
Peptamers.
DNA or RNA keys.
Exactly. Single strands of DNA or RNA that fold up into unique 3D shapes that can bind targets very specifically, much like antibodies. But they have advantages. They're smaller, so maybe better tumor penetration.
Less likely to cause an immune reaction. Generally, yes, lower immunogenicity. Easier to make, often more stable. People are quite excited about aptamers. PSMA aptamers for prostate cancer are a good example again.
Any other major keys in this targeting toolkit?
Yeah, a couple more worth mentioning are folic acid and transferrin. Folate receptors are often overexpressed in many cancers, so folic acid is a simple, low-toxicity targeting agent. And transferrin targets the transferrin receptor, which cancer cells need a lot of because they're metabolically active. A bonus with transferrin is that it can naturally cross the blood-brain barrier.
Oh, that's huge for brain tumors, right? Which are notoriously hard to treat.
Exactly. It potentially opens a door there.
Okay, so we have these smart, triggered, targeted nanoparticles. What are they actually carrying? What's the payload in this smart missile? What's the ultimate goal here?
The ultimate goal is simple but profound. Better cures, fewer side effects, longer lives for patients. That's the bottom line. And the payloads, the stuff inside that toolbox, have definitely evolved.
I'm guessing it started with standard chemo drugs.
You got it. Initially, it was mostly about delivering small molecule drugs, the traditional chemotherapies. Nanoparticles were great because they could help dissolve drugs that weren't very water soluble.
Right.
And crucially, by targeting, they could reduce the awful side effects. The classic example everyone points to is doxyl liposomal doxorubicin, FDA approved way back in '95, first nanomedicine approved. It really showed the potential.
A foundational step. But treatments have moved beyond just small molecules. What about bigger things?
Definitely. Next came proteins and peptides. These can be very potent, very specific, but they're often fragile in the bloodstream or the body attacks them as foreign.
So the nanoparticle protects them.
Exactly. Protects them from breaking down, shields them from the immune system. Using things like pegylation, adding a polymer coat, helps them last longer in circulation.
Okay. And then came the gene therapy revolution, right? Delivering nucleic acids like RNA or DNA. That must be tricky.
Super tricky. Nucleic acids like siRNA, mRNA, they have amazing potential to, turn genes on or off. Silence faulty ones, boost helpful ones. But on their own, they get chewed up by enzymes instantly.
Useless if they don't get there.
Right. And getting them inside cells is hard too. Nanoparticles, especially lipid nanoparticles, are essential. They package, protect, and deliver. Onpattro, the first approved siRNA drug, relies on lipid nanoparticles. It really opened the door for genetic medicines.
Now, this next one sounds like pure sci-fi. Delivering living cells. How does that even work?
It sounds wild, right? But it's a very active area of research. The idea is using smart nanoparticles, perhaps as carriers or support systems, to deliver living cells like FAR T cells.
The modified immune cells that hunt cancer.
Exactly those. Getting enough FAR T cells into a solid tumor and keeping them active there is a huge challenge. Nanoparticles might help guide them, protect them, maybe even provide signals to boost their activity right inside the tumor microenvironment.
Wow. So enhancing our own immune fighters.
Precisely. It's about boosting adoptive cell therapy. Still challenges like cell dose and getting through dense tumor tissue, but nanoparticles offer new strategies.
And it's often not just one drug, is it? Combination therapy is key for cancer. Can nanoparticles handle multiple payloads?
Yes, and that's another major advantage. Smart nanoparticles are fantastic for combination therapy. You can load multiple drugs into the same particle.
Why is that better?
Because you can ensure they arrive at the tumor cell at the same time, and maybe even in a specific optimized ratio. This can be much more effective than giving the drugs separately, especially for overcoming drug resistance.
Is there an example of that in practice?
Absolutely. Vixios, or CPX351. It's a liposome that carries two chemo drugs, cytarabine and daunorubicin, in a very specific 5-to-1 ratio. It's approved for a type of leukemia and significantly improves outcomes compared to giving the drug separately. Shows the power of co-delivery.
This is incredibly sophisticated stuff. Where does AI, artificial intelligence, fit in? You mentioned it as the blueprint for the next generation.
Yeah, AI is poised to be a complete game changer here. It's like giving our nanoparticle designers superpowers. AI can process data and find patterns way beyond human capacity.
So designing better nanoparticles. Wow.
Several ways. First, enhance drug delivery. AI can analyze incredibly complex patient data genetics, tumor characteristics to design nanoparticles with truly intelligent navigation.
Tailored to the individual patient's tumor.
Exactly. Selecting the best targets, predicting how the particle will behave in that specific tumor environment, truly personalized. Some are even exploring concepts like using AI with, say, tiny drones or microrobots for delivery in the future.
Mind-blowing. Okay, what about the actual design process? Can AI speed that up?
Massively.
Yeah.
That's the design and optimization part. AI algorithms can simulate countless variations, size, charge, coding, drug loading, and predict which designs will be most effective before you even make them in the lab.
Saving time and resources.
Huge amounts. It dramatically accelerates discovering and refining new nanoparticle formulations, finding the needle in the haystack much faster.
And once the nanoparticles are in the body, can AI help track what they're doing?
Yes, that's the real-time monitoring aspect. Imagine nanoparticles equipped with tiny sensors or imaging agents. AI could analyze the signals coming back.
Like getting live updates from the battlefield.
Kind of.
Right.
Monitoring how the tumor is responding, are the drugs working, are there side effects developing. This allows for adaptive treatment, adjusting the dose, changing the strategy on the fly based on real-time feedback, a closed-loop system.
Truly personalized and dynamic. Could this tech also help catch cancer earlier?
That's another huge area of promise. Early diagnosis. Think of nanosensor arrays, tiny sensors that can detect minute changes in biomarkers in blood or breath.
Ah,
Combine that with machine learning AI to recognize complex patterns, disease spectral fingerprints unique to early stage cancer. There's research showing this can distinguish, say, ovarian cancer patients from healthy people with amazing accuracy, potentially much earlier than current tests.
Okay, this all sounds incredible, almost too good to be true.
Mm.
What's the catch? What are the big hurdles keeping smart nanoparticles from being standard treatment today?
Yeah, despite all this amazing potential, the sources are clear. We're still at the relatively preliminary stages of clinical applications. There are major roadblocks.
Like what?
Two really stand out: safety and toxicity. This is non-negotiable. We need exhaustive testing.
More than just checking if it kills cancer cells.
Way more. We need to know what happens to these particles in the body long term. Do they build up in the liver, spleen, kidneys? Our sources highlight this often. Nanoparticles accumulate in vital organs rather than being safely cleared.
That sounds like a serious problem.
It's a huge challenge. Potential long-term toxicity from this accumulation is a major barrier. And we also need to understand immunotoxicity. Do they trigger harmful immune reactions? It requires incredibly careful evaluation.
And then there's the regulatory side. Getting approval from agencies like the FDA must be a nightmare for something so new.
You nailed it. Our sources call regulatory approval the main obstacle. The process is just incredibly long, expensive, and complex.
Especially if the rules haven't caught up with the tech.
Exactly. There often aren't specific guidelines for nanomedicines, which complicates things. Think about it. Doxil was approved in 1995. Despite decades of research since then, only a handful more smart nanoparticle cancer drugs have actually made it through FDA approval.
Wow, only a handful. That really puts the challenge in perspective. It sounds like a very slow climb.
It is, but it's not hopeless. We have had successes like Abraxane, like Vixios we mentioned. They prove it can be done.
So there are signs of progress.
Definitely. And there are numerous smart nanoparticle systems in clinical trials right now, testing different approaches, different targets. The pipeline is active. The research is pushing hard. There's real, tangible promise, even if the translation to the clinic is slow and difficult.
Okay, so we've unpacked this amazing toolbox of smart nanoparticles. We've seen their potential for intelligent response, precision targeting, carrying diverse payloads, and this incredible boost from AI. It really does feel like a leap towards that tailored suit for fighting cancer.
It really does. The potential to improve cures, reduce suffering, extend lives, it's immense. But, we have to keep our feet on the ground about the hurdles. Safety validation, that long-term accumulation issue, navigating the regulatory maze. These are serious challenges that need solving.
A field full of incredible science, but also real-world complexities.
Absolutely. Balancing the huge promise with the practical realities.
Which brings us to a final thought for you, our listener. Picture that future. Cancer treatment not as a blunt instrument, but as that perfectly tailored suit designed by AI, delivered by nanoparticles, constantly adjusting to your specific biology. It's an amazing vision. But think beyond the lab. What else needs to happen? What changes in healthcare systems, in policy, maybe even in how we think about medical innovation would be needed to make that truly personalized future a reality and accessible for everyone? Something to ponder.